Okay, well, thank you, Jerry, for the invitation. I always love to hear about groups that want to know more about propolis and understand more about the benefits of propolis to honeybee health. Um, I find this topic fascinating, of course, because I dedicated a lot of years and of my life in studying propolis. So I find fascinating. So I love to hear when people also want to know more about it. So it's a pleasure for me to talk about it with you guys today. So I titled this talk as "The Benefits of Propolis to Honeybee Health," because that's was the main focus of my research. My PhD research was to understand how propolis benefit not just the honeybee itself, the individual, but also colony health. One way that I like to see as the immune defenses mechanisms in honeybees is not just the individual immune system, but is a very complex way because honeybees are social insects. So they rely not just on their individual immune defenses, but they also have their other mechanisms of defenses that we call their natural defenses, their behavioral defense mechanisms. So it's a very complex way on how bees maintain health in a colony. If we think about humans, for example, one of our main defense against pathogens is our immune system. But we also have behavioral immune defenses, natural defenses that we learn right now, which are washing up. So some behavior, washing our hands and maintaining those social distances, right? So there are also behavioral defenses that sometimes we don't really pay attention to it, but in honeybees, just because how the society works, we really have to to understand and be able to take that all in so we can better work with bees and better help them to maintain that health. So bees have their immune system just like many organisms, so they do rely on their immune system to maintain health in a colony, but they also use chemical secretions. They have spatial organization, physical barrier, behavior defenses, and maybe others that we haven't found out yet. So it is very complex, and we can make some similarities to humans as well as we go. And I find many times easy to understand bees when we can relate to something that we understand much more, which is our own maybe immune system. So just keep that in mind as we go. That is very complex, but we'll try to make similarities here and there to understand. So bees have their individual immunity, but like I mentioned, because they are a social insect, they have their social immunity as well. And these two terms have been used in the science world very often. So we call the individual immunity and the social immunity. The social immunity term, I think, was coined in about 2006, I think, was the first paper explaining behavior differences as social immunity. So I really like that wording. I think it's brilliant to call behavioral defenses as our social immunity. So in honeybees is the same, and their individual immunity is their individual immune system. So that is for honeybees because they're insects. That it could be cellular immunity, which is the encapsulation of pathogens by hemocytes. Hemocytes are like blood cells. So those cells will encapsulate any pathogen that anything that they recognize as a pathogen. So they encapsulate and they kill it that way. But we humans, we have antibodies. Bees are insects. They don't produce antibodies, they produce antimicrobial peptides. Those are proteins that are also able to kill, to identify and kill pathogens. So anything that they recognize as a pathogen. And their immune system, we have hygienic behavior, which some of you may be familiar with. That is a behavior that some bees express in the colonies, and they're able to detect much early that a brood is infected with a pathogen, a brood is not healthy, and they will detect before that pathogen become infectious, and they will remove from the colony that brood that is infected with a pathogen. So we call that a hygienic behavior when a bee is able to detect a pathogen from a brood much early before it becomes infectious. There's also grooming behavior, which I'm sure most of you are familiar with as well. When bees remove, let's say, varroa mites from their own body or from a nestmate's body. And as the focus for today will be collection of plant resins. That is also considered a social immunity behavior. And I will explain here why the collection of plant resins is considered a social immunity behavior, which hasn't been studied very much, but as we will go through today, it's a big part of their social immune system. So plant resins are produced by trees. It's mainly produced in young leaf buds. So the production of resin is the main defense mechanism of a plant. That is how they defend themselves from herbivores and pathogens. So plants usually will coat young leaf buds with resin, and that protects that young leaf bud that is very vulnerable from herbivore attack and pathogens. So it's rich in antimicrobials. So it's a very important defense mechanism of the plant. They will also produce resin when they have been attacked. So they can also induce resin production. If there is a herbivore attack, they will produce a localized resin where they can at least protect that part of the plant that has been attacked. So we have the induced resin production and the resin production that occur regardless of the presence of pathogens, which we can find in young leaf buds. So the trees is constantly producing resin. It is rich in antimicrobial compounds, and not just bees, but other animals as well use resins for their own health benefits. So we find this among other animals in the animal kingdom, and bees is one of them that can use resin for their health benefits. So bees will collect resin from plants, and they will use their mouth parts to collect the resin, to scrape the resin from the plant, and they transfer that resin.
If I can get this on the TV.
That they collected using the mouth parts, they'll transfer to their pollen baskets, as you can see here. So the same way that they transport pollen, they transport resin using their pollen basket. And when they get to the colony, they will deposit that resin. Often they'll deposit along the hive wall. So the sides of the colony, if they're nesting in a tree cavity, we can see that they deposit all along the wall of the colony where the colony is nested. So you can see this red line here. It shows where the colony is started and where the colony was not nesting. So above the red line, the colony was not nesting, there was no comb, and below the red line, the colony was nesting. So you can see that it's very dark, and that that brown color that we see, that's the propolis deposition. So that's where bees usually deposit propolis in a natural tree cavity. They will also deposit where the comb attached to the tree, so where it makes that connection. They will also make like an antimicrobial barrier right there where the comb is attaching to the tree. So it's really nice that we can see that distinction because it shows us how bees prepare their nest, that they line that area with propolis. So I just have a few more photos on bees collecting propolis. And as you can see here is on their corbiculas, on their pollen basket. And one main thing that you can tell that is resin and not pollen is because it's not a regular shape. So it's not that beautiful pollen ball that is nice and soft and circular. It has many points. It's very irregular and it's very shiny. So that's how you can tell that it's definitely resin that they have there and not pollen. And so you will see they're coming into the hive with the resin on their pollen basket, and its resin is very sticky. So it's very difficult for bees to remove that resin from their hind legs on their own. Usually with pollen, when bees get back from the hive with pollen in the pollen basket, it's fairly easy for them to scoop it out or to have a help from another nestmate to scoop that pollen out into a pollen cell and have it stored there. But with resin, it's very difficult because it's very, very sticky. In average, a pollen, when they come back with pollen, to remove it and store it away is about 1 minute. With resin, it can take about 30 minutes for them to get the resin unloaded from their hind legs. So it can take up to 30 minutes, and that's a lot of time in the bee life. So it's not an easy task, but they still do it. So in my opinion, if they still do it, even though it's not that easy, it's because it is important to them. So when they get back to the colony with resin on their pollen basket, they need the assistance from another nestmate to remove that resin from their pollen basket, as you can see here in this photo. And the nestmate will use its mandible mouth parts to remove that resin from the hind legs. When I was doing an observational study, looking at specifically at resin foragers, I saw a lot of resin foragers that were missing one hind leg. And my guess is because sometimes it sticks so hard to their hind leg that I think when they are getting the resin removed, I think they might have the hind leg also removed. When I was collecting resin from their hind leg using an insect pin, I a few times also removed their hind legs with the resin load, which made me very sad. So, and I do find that quite a bit in a colony when they're coming back with resin. Sometimes they only have one leg and they still have resin on that leg, but it's just very, very interesting. So it's a very difficult task, but they do it. So the nestmate that remove from the hind leg using the mouth parts, they will then go to the place in the colony where it needs resin to be deposited, and they will deposit in that area. So here, as you can see, the worker bee is depositing propolis on the top of the frame. So here I have used two terms, propolis and resin. And many people use them as if they are the same thing. Technically, they are sort of the same thing, but resin we consider as a single source product, right? It's a product from one specific tree. Propolis, it's when they are inside of the hive, and it's a combination of many different resin that bees have collected from all the trees that they have available. So propolis is more a mixture of resin. Um, there is some mandibular secretion also found in propolis, whether it is on purpose or just because of how they manipulate propolis, we don't know. But because they use the mouth parts, we just assume that is due to the manipulation of the resin and the propolis. But we also find wax in propolis. So resin is a tree product. Propolis is a bee product. It's a mixture of many resin with some mandibular secretion and also some wax. Resin in, well, propolis has been studied for many years, but mostly because of the human health benefits. You can find propolis, studies about propolis from many, many, many years ago, and all of them are related to human health and how propolis can benefit human health in many different ways. But only recently we started to uh, take a look on how propolis benefit honeybee health, which to me is quite amazing because propolis come from honeybees, and yet we first studied the benefits of propolis to human health and never looked into the benefits of propolis into honeybee health, and mostly why they collect it, right? So if it's in the hive, there is a reason for that because it's not easy to collect. There is a lot of time, it's a time consuming task. So there must be a huge benefit for bees to collect it. And yet only recently we started to look more into this and deep dive on the benefits of propolis to honeybee health. And today I will talk not just propolis, but something that I like to refer as the propolis envelope. So if you remember the photo that I showed before of that tree cavity with the red line and that dark brown where the colony was nesting, uh, that is what I call the propolis envelope. That's because the bees, usually in a natural tree cavity, they will deposit propolis surrounding the nest where they're located, and that's why we call it that propolis envelope because it's all surrounding their nest, and that is in a natural habitat in a natural tree cavity where they are nesting. But when we're using man-made boxes, like in this photo, we don't see that propolis envelope. We do see propolis in cracks and crevices where the frames sit on the box, like you can see here on top. We do see that they deposit propolis there. And some hives may deposit a little bit more propolis than others, but we still don't see the same propolis envelope that we see in a their natural habitat, in a tree cavity. Like this one, for example, as you can see the big difference. One thing that I like to kind of point out is that we have taken bees from their natural habitat, right, in a tree cavity, and we have domesticated bees inside of these wooden boxes. And by doing so, we ended up removing one part that I believe is an important part of their nest architecture. That is the construction of this contiguous propolis layer that they build when they nest in tree cavities. Because these are on the left in man-made boxes that what we see more often. That is propolis only deposited in cracks or where the frames are sitting or between frames kind of thing, but not that propolis envelope. So one thing that I wanted to create in man-made boxes, so in our regular beekeeping equipment, was a propolis envelope. So I could study uh, the effect of a propolis envelope. I had to create that environment because it would be really difficult for me to study them and in a controlled way, them in their natural habitat inside of a tree cavity. It would be really difficult for me to make manipulations and sample collections and have a very standardized way across experiments. So I had to do my best to recreate that environment in our regular boxes. So to do so, I use propolis straps that you can buy. Those are commercially available propolis straps. And I put it, staple them on the four walls of the box. So I cut them to the specific size and staple them to the side of the boxes to stimulate the bees to fill each crack of that propolis strap with propolis and then create that propolis envelope. And this is what I had at the end. So this is a three boxes, one on top of each other. And you can see that pretty much all of the gaps of the propolis strap were filled with propolis. So I was able to somewhat create that propolis envelope that they have in a tree cavity inside of our commercially available beekeeping equipment. So I could study the effect of that propolis envelope. So I looked at one thing that I looked was the individual immune system. So like I mentioned to you guys before, honeybees have two different immune systems, right? The individual immune system and the social immune system. So I wanted to look at also their individual immune system. And one part of their individual immune system is the antimicrobial peptides secretion, which is kind of like our antibodies, if we can make a fair comparison. So it's like the bees' humoral immune response. So I looked at 6 different antimicrobial peptides, and I saw that when the propolis was present, the bees that had that propolis envelope, so these are the red bars, those are the colonies that had a propolis envelope are the red bars, and each of these here are antimicrobial peptides. So I look at 6 different important proteins that are part of their immune system, the individual immune system. And when the colonies had the propolis envelope, that protein was way less expressed. So, which means basically all our immune system has always some level of expression because we have bacteria in the air. We always have some pathogen that our body is fighting. That doesn't mean we're sick. It just means there's bacteria or fungi everywhere. So our body always is fighting something. So our body is always expressing some level of in the honeybee world, antimicrobial peptides. There was always some expression. But when they had the propolis envelope, they didn't have to express this much, was much lower. So we say that their immune system was quieter, was they didn't have to invest as much energy to keep that immune system active. So it was much quieter for when they had that propolis envelope. If you think of your regular immune expression as a baseline expression, because like I said, we always fighting some bacteria or fungi that is in the air or it's in contact. That's your baseline expression. It will always exist. But if you can lower that a bit, because maybe you coat your house with propolis, or you always washing your hands or something, you are able to lower that baseline expression, you're saving some energy. Our immune system, honeybee's immune system, it's a very costly system. It takes them a lot of energy to produce and to keep that system activated. So if they're able to lower their immune system, that baseline expression, a little bit, they are saving some energy. So their immune system is quieter when they have that propolis envelope. We also noticed that bees that had the propolis envelope in 1 year, the colonies that had propolis envelope, in 1 year, it increased survivorship over winter survivorship. The second year was not different than the control colonies, but at least on the first year, it increased their survivorship, and also brood population size in the spring. So they had the propolis envelope in the summer, in the fall, and they had somewhat similar brood population size. But in the following spring, after the winter, they came out of the winter much stronger. They had the brood population size was higher than colonies that did not have that propolis envelope. But those were bees that were healthy. We didn't challenge those bees with any pathogen. We didn't manipulate them in any way in terms of pathogen or diseases. Those were bees that were healthy, and we checked their pathogen level. So we checked Varroa, we checked Nosema, we checked different viruses, and they were all similar from the bees, the colonies that did not have the propolis envelope, and the colonies that did have the propolis envelope. So they were considered healthy colonies. So in healthy colonies, we noticed that the propolis envelope had benefit in their individual immune system, and possibly also by having stronger colonies in the spring with higher brood population. But what about when you challenge them with a pathogen? So when they come in contact with a pathogen, when they have a pathogen infection, what are then the benefits of the propolis to that hive? So I'm infected some colonies with Panibacillus Larvae, which is the bacterial pathogen that causes AFP. This is a brood disease that can only infect 1 to 2 day old larvae. That's the susceptible stage. If an older larvae becomes in contact with that bacteria, it will not die from it. They have to come in contact with this bacteria much early in their brood life stage. So 1 to 2 days old. The larvae comes in contact with the bacteria via oral intake of contaminated larval food, or the larval food gets contaminated, let's say with the spores, because the spores were already in the cell from a previous larvae that died from that pathogen, and those spores remain in the cell, and that brood happened to be raised in a contaminated cell. So it's either from contaminated food, or because the bacteria is already in that cell where that brood was raised. One mechanism of resistance to AFP is associated with larval food antimicrobial activity. So some studies have shown that one mechanism that larvae can be resistant to these bacteria is when the larval food that is presented to the larvae has higher antimicrobial activity. So this was a study, I think, in the 60s, where they looked at different genetic lines and different larval food presented to those genetic lines. And the larval food in genetic lines that were resistant to AFB, had an antimicrobial activity that was much higher. So what I did to study what would the benefits of propolis be when the colony was challenged with this bacteria, is that I set up a few different treatments where some had the propolis envelope and were also challenged with the bacteria. Some had the propolis envelope, but they were not challenged at all. And a couple of other treatments did not have a propolis envelope, and one was challenged and the other one was not challenged with the bacteria. So we had 4 different treatments, so I could understand the 4 different scenarios and compare among these treatments. I sprayed the colonies with the bacteria to infect them, to challenge them with this bacteria. So you have to spray these colonies at the right brood stage, because again, they're only susceptible very young. And so I like to say that sometimes when we're studying bees and we're doing research, we do the reverse beekeeping. Instead of keeping colonies healthy and trying them not to get AFB, we spray them with AFB so they can get very sick. So we do a bit of reverse beekeeping to be able to understand how colonies defend against this pathogen. So I had to spray them with AFB and then check pretty much every day to see signs of AFB, which you can see here in this figure. Signs of AFB, you can see a brown brood, and spray them one more time, collect samples for the larval food, because as I mentioned, it's one mechanism of resistance to AFB is the antimicrobial activity of larval food. So we collected that, checked again for signs, and then finished the experiment in October. One thing that I found, so I looked at these bees and I looked again at their individual immune system. So I looked at antimicrobial peptides. In this case, I looked at 3 different antimicrobial peptides. And these are proteins that have shown to be important in the defense against this bacteria. And I looked at the symptomatic period, which means that it's after they show signs of the disease. And I found the opposite that I found when they were healthy. When the colonies was healthy and they had the propolis envelope, if you remember, I said their antimicrobial peptides activity was lower. So they were saving energy. So the baseline was lower. When they were challenged, the colonies that had propolis envelope and were challenged, that antimicrobial peptide activity was higher. So it was much higher than the colonies that were also challenged but did not have the propolis envelope. And I looked at larval food and looked at the antimicrobial activity of the larval food because we think it's an important mechanism of defense for this pathogen. So I collected the larval food, took it to the lab, and in the lab, I was able to measure the antimicrobial activity of that larval food. And one very interesting thing is that the larval food from colonies that were challenged with the bacteria and had the propolis envelope was much higher. They were able to inhibit the growth of the bacteria much better than larval food from colonies that did not have the propolis envelope. So this was very interesting. So here it's before they show any signs of the disease, the antimicrobial activity of of the larval food from colonies that were challenged and had the propolis envelope was much higher than every single treatment. After they had the signs of the disease, it was only significantly higher than colonies that did not have the propolis envelope and also were not challenged with the pathogen. And the other two treatments were intermediate. I know this looks lower, and I'm saying higher because it's showing the growth of the pathogen. So there's much less growth of pathogen compared to the other one. So it's less growth of the pathogen. So it means higher in activity of the larval food, basically. Um, so one question, like sometimes we run these experiments and we end up with more questions than answers. And one question that we had was what the main thing is, why is this larval food higher in antimicrobial activity compared to all the other colonies? And one thing that one of our hypotheses is that maybe the nurse bees that are synthesizing more antimicrobial peptides are able to incorporate these peptides into the larval food. So I mentioned that one of my results was that the antimicrobial peptides were higher, right, in colonies that had the propolis envelope and were challenged compared to colonies without propolis envelope. So they had a higher expression of antimicrobial peptides and also a higher antimicrobial activity of the larval food. So this is one hypothesis, that they synthesize more antimicrobial peptides and that they also incorporate those peptides into larval food to give some mechanism of defense to that very young larvae. And the other hypothesis is that maybe volatile compounds from propolis, because propolis is a mixture of resin, and resin has many antimicrobial compounds. So maybe some volatile antimicrobial compounds from the propolis are also found in the larval food, because those are volatile compounds. Maybe they can also contaminate in some way the larval food and give that larval food its higher antimicrobial activity. So these are two hypotheses, and we did not run any experiment to test these hypotheses, but those are questions that we kind of ended up with at the end. One thing that I also measured was the visual signs of the disease. So AFB, American Fall Brood, is very easy to assess in the field and count how many cells have that disease, because you can see the dead larvae. You can see the AFB scales. As you can see in this photo here, for example, these two cells have AFB scales. And it's fairly easy to identify in the field and keep a record of how many cells in a single frame were infected with AFB. So how many cells have AFB scales. So we did that in the field and counted in every single colony how many AFB scales were found in the whole colony. And in August, colonies with propolis had less AFB scales compared to no propolis, but they were not significant different. So numerically, they were lower, but statistically, they were not different. So the same thing we found in September, they were numerically lower, but still not statistically significantly different. But in October, 2 months after we had started with our challenge, with our bacteria challenge, we found a significant difference between colonies with propolis and colonies without propolis. So that trend of having always lower was significantly different, statistically significant different in October. So colonies that had that propolis had significantly less cells with AFB scales compared to colonies that had no propolis at all. So this doesn't mean bees with a propolis, colonies with a propolis envelope will be able to clear AFB from the colony, because we did not observe that. They still had AFB scales. AFB was still there, so it did not clear, but it was significantly lower. Maybe because it might help them to lower. Maybe they will be able to deal with that on their own. We don't know, and I cannot guarantee it will. But at least we did see that it was a lower infection compared to colonies that had no propolis envelope. And then just to end this, one question we had was, okay, propolis seems to be very important to colonies, to honeybee colonies, because when they're healthy, it seems to benefit their immune system by keeping the baseline of expression quieter. So bees do not have to allocate as much resources to maintain their immune system as bees that do not have a propolis envelope. So if they do not have to allocate as much resources to maintain a high expression immune system, they could use those resources in other things, doing other tasks in the colony. So they're saving energy here, and maybe they're able to do more elsewhere, let's say. So when they're healthy, it seems to help them in that way. And when they're challenged with a pathogen, the presence of the propolis envelope seems to help them to fight a pathogen much better, because we saw that these colonies had a higher expression of those antimicrobial peptides. So they were able to fight a pathogen much stronger, let's say, because the expression was higher than colonies that did not have a propolis envelope. The antimicrobial activity of the larval food was better, was higher. So they not only could express their individual immune system better, higher, they also seem to be able to protect the larvae and protect the brood better, because the antimicrobial activity of the food was higher. So if it's that important for the colonies, then we kind of assume that bees, we kind of like to make that comparison with humans, that we go to the drugstore and we get the medications we need to fight some pathogen. Bees don't have that luxury, so they kind of using resin as their drugs to fight pathogens. So instead of going to the drugstore, they go out into nature and they collect resin, so to medicate. So they're self-medicating maybe when they are infected or challenged with a pathogen. So we wanted to study that self-medication aspect of the propolis, if they use resin to self-medicate. So that was one question that we asked at the end, if bees self-medicate in response to infection. So we used two different pathogens. One was a bacterial pathogen, so the same one, the Panibacillus larvae that caused AFB. And we also used another pathogen, a fungal pathogen that causes chalk brood, to study the responses to two different pathogens. For that, we had to sit in front of colonies and count every single returning forager for 15 minutes twice a day, and then count how many of those foragers were resin foragers. So basically, what you do is you have your colonies and you close the entrance, and for 15 minutes twice a day, you counting all the returning forages on that 15
Uh-oh, I don't know what's the problem here. There may have been a outage.
Or the weather is freezing her internet.
That's too bad. This is like one of the most interesting parts.
Very interesting, huh? Where is she located?
Fantastic talk.
You said Alberta,
Alberta.
Canada.
Mm-hmm.
Welcome back.
internet my internet failed. I can share my screen again, Jerry, but you might have to give me host
Yeah,
privileges.
just a second.
Yeah, no
Okay,
problem.
go ahead. Welcome back.
Thank you. The internet went down, but it's back up. Okay, so I think I was here. So yeah, so this was for the self-medication. So basically, we measured the before and after pathogen challenge, and if the measurement of the number of resin forages after you challenge is much higher, that means that you're self-medicating in response of that pathogen challenge. So we did find, and this was pretty amazing, that bees self-medicated very strongly for one pathogen, which is the fungal pathogen, the chalk brood, but not so much for the bacterial pathogen. So they seem to self-medicate more in response to chalk brood infection than AFB infection. Numerically, it was higher than colonies that were not challenged, so those are called the control. But statistically, they were not significant. They're just numerically higher, but not significant. So we cannot say that they self-medicate for AFB, but they definitely self-medicate for chalk brood, which is interesting, and it raises a bunch of questions of why they self-medicate for one pathogen, not the other. There's many hypotheses for why that could be possible. One that I like is that insects and animals fight pathogens, different pathogens in different ways. So different pathogens will have different pathways on how they infect the organism, and because of that, that trigger different defense mechanisms. So maybe with a fungal pathogen, it triggers different pathways and triggers that specific defense mechanism, that is the collection of resin for fungal pathogen. But with bacteria, specifically that one, the P. larvae for American fall brood, because it triggers different pathways, different brood life cycle, their defense mechanism is different. And maybe their defense mechanism for honeybees is not through self-medication, not that strongly at least, but they might allocate more resources into different defense mechanisms and not self-medication. So that was very interesting, but again, it raises a bunch of questions and answers a few, but it raises more questions at the end. So just to kind of wrap up, what we can learn from all of this is that bees, they don't rely on a single layer of defense mechanisms. So they don't only rely on their individual immune defense, which is the um, synthesis of antimicrobial peptides, let's say. They have multiple layers of defense mechanism. So one, let's title that as their individual immune defense. The other one could be their social immune defense. So what we saw with the larval antimicrobial activity. So they have another layer of defense that is a behavioral defense of protecting the brood, protecting a vulnerable brood, that very young larvae. So that's a defense, behavioral defense. But they may also have another layer of defense that is with that Propolis envelope, that is an architecture critical for that social system, is an antimicrobial layer, is an antimicrobial layer that protects not just the individual immune defense, but it may also protect the colony health in general, the brood, and the adult as well. So it's multiple layers of defense, and when we study social insects, we cannot just look at the individual itself. We have to kind of look the whole picture and understand how social insects defend the colony, and in every single layer of that defense mechanism that is important for the total, basically. So this research was funded by several institutions, and there was many people involved that helped with this, and donations and beekeeping organizations that also assisted, and as well as many collaborators to make all of this happen. So I'm very thankful to all of them. I did ask Jerry to ask the group a few questions so I could be able to answer some. So I have a few questions here that I received from Jerry on the email. So I'll try to answer some of them, and then we can, if we have time, we can open the floor for more questions. Maybe I did answer already some, but I'll go through. So the first one, what solvents or combinations of solvents work best on propolis? Alcohol plus something else. So resin, the best solvent you can use is alcohol. You can use water, so you can do a water extraction of those antimicrobial compounds. But the best way to extract those antimicrobial compounds is by using alcohol as a solvent. And we have tried water, but you don't extract as many antimicrobial compounds in a water extraction as you can extract with an alcohol extraction. So I would recommend alcohol. You don't need to add anything else. You can just add ethanol, and that would be a good solvent to extract those compounds, basically, that are in the resin. What plant families in North America do bees get propolis from? Bees get propolis from pretty much any kind of plant that produce resin. There was a study in 2015 in Minnesota that they looked at in a 5-mile radius what was the plant source that the colonies were collecting from, and it was pretty much everything, all the trees, the resinous trees that was available. There was pine trees, there was poplar, there was everything. Bees seemed to, let's say, favor. I don't really like to say they use that word, but they favored some trees specifically. So we noticed that bees tend to collect way more resin from poplar trees. So that's one tree family that they do collect a lot of resin from, is poplar trees. So you can see your cottonwood or just balsamic poplar. I think they also collect a lot from that one. So, but most poplar, oh, poplar, that family, they collect a lot of resin from. Some medicinal aspects that there is many, especially for human health. There's a lot of work on the propolis activity on human viruses, human bacteria, fungi, and everything. And with honeybees, like I said, there's not a lot of work on that, but we were able at least to look at self-medication for 2 different pathogens, and other people have looked at self-medication for chalkroot as well. And I think there is a group that looked at self-medication for nosema as well. So there is a little bit of work done on that area. What info is out there that's false? I don't know. I haven't come across a lot of false information about propolis. I guess mostly because there isn't a lot out there about propolis. So I'm not. I don't think there is a lot or any that I know of. Should we save our propolis scrapings? What should we do with them? You can save propolis scrapings, and you can do ethanol extractions. That is, I think, the main thing you can do with propolis. Is a lot of people do is ethanol extractions. A lot of people also use propolis in hand creams. So especially in the winter, it's really nice. I have one. It was even here my desk. It's a hand cream with propolis. It has been used in the cosmetic, I guess, industry quite a bit. So if you are into that kind of stuff, you can make some of that soap and so on. But for honeybees specifically, if you want to use it back in the hive, you can do an ethanol extraction. Some people have used that to coat the inside of the hive with an ethanol extraction. The note I would say on that is that we don't know how long that effect lasts when you coat your hive with an ethanol extraction of propolis, because the studies on that have only lasted 7 days after application of that ethanol coat. So we are only able to tell you within that 7 days what the effect of that ethanol extraction is. Past that, we don't have any long-term studies that have looked into that. So with my research, I looked at the natural deposition. So I looked for 1 year, and there was still effect, right? But that's a natural propolis deposition, is different than an ethanol extraction of a volatile solvent. So we don't know how long that will last. What effort have proven successful in getting bees to coat the inside of hives with propolis? I have tried window screen with not much success. I have also tried window screen. Giraffe textured boxes work. I have tried in window screen. It also didn't work. It's not. It does not provide an enough rough surface for bees to encourage, to trigger that resin deposition. So bees, they will dance and recruit for honey, for nectar, I should say, and recruit for pollen, as well as they recruit for resin. So bees, they sense the need for resin in some way. There's not a lot of studies that have looked in very much detail on what necessarily trigger that resin collection, besides what we looked at, self-medication, for example. But something triggers the need for resin in the colony, and bees will recruit for more resin. And the roughness of the surface seems to stimulate that resin collection. As we can see inside of a tree cavity is very rough, and that triggers resin collection, triggers them to fill those gaps and create that smooth surface, right? And the window screen is not rough enough to trigger that resin collection. So I would try roughing it up, the side of the box, the inside of the box a little more, but make it deeper grooves than just the size of like a window screen kind of thing. So I use straps, propolis straps, and that depth really worked. So I would try something that deep. Should we be selling propolis collection, or put another way, how do we investigate propolis collection? If you want to instigate propolis collection, you have to the best way is to give them a rough surface, and that will stimulate. Yeah, I would say that's the best way. Or you can challenge your colonies with some pathogen, and that seems to work as well, but I don't think you want to go that way. Is it true a pinch between the cheek and gum will ward off evil spirits, or at least push off a cold? I'm not sure if I understand that question. I will skip, but maybe whoever asked that question can ask me again.
Yeah, so if you take some propolis out of the hive and just put it in your mouth, does it have any benefits to you straight away?
Oh, I see. I don't know. I don't think I can answer that. There has been lots of studies, like lab studies, on the activity of the antimicrobial compounds from propolis in different bacteria and even viruses. So if we're talking about a flu, which is a virus, there has been studies that show that some antimicrobial compounds from some resins have a high activity against human viruses. But I don't know if there has been a lot of actual human studies on that. So I personally use, and I like it, but I wouldn't be able to answer with certain that question.
Now, the more interesting part of that question is warding off evil spirits.
Yeah, that would be cool. What is the recommended way to use propolis as a wound dressing? Again, I don't think I am able to give a recommendation on that. Personally, I just spray propolis extraction on my finger if I have a cut, like if I'm camping or something, and you don't take a lot of medication with you. A lot of people take a bit of honey. I have propolis in a spray bottle. I don't think I have any here with me, but having a spray bottle, so I take that.
Is the spray suspended in ethanol or is it water?
Mine is in ethanol. I have bought some in water for my nieces, because they're fairly young, and I don't want to give them alcohol just yet. Just kidding. But it's in water for them. But I have some for myself, alcohol. I prefer. I think it's a much better solvent.
Doesn't that burn a cut?
Oh, yes, it does.
Yeah, but you get used to it though.
It also sanitizes the area with the alcohol.
Exactly.
Double duty.
I'm thinking that's closer to the warding off the evil spirits thing there.
Yeah. Because I use 75% ethanol in my extractions. So it's a fairly strong ethanol.
And then you just filter out whatever debris is in there?
Yeah, you can use cheesecloth. I use coffee filter.
Coffee filter. Mhm.
Yeah, I use coffee filter, and the way I do, I use a glass bottle. So you want to use glass, not plastic, when you dealing with solvents. So you put your propolis scrapes from the colony in a glass bottle, and you fill that with ethanol. You can adjust the percentage out to however strong or weak you want your extraction to be, 10%, 13%, or something like that. So you
By weight.
add, yeah, volume to weight. And so you adjust the amount of ethanol you need to add according to the propolis you have. And in a glass bottle, and it's better to use a dark glass bottle, just because, again, you're using solvents. So you want to have it also protected from the light. So you use a dark glass bottle, and you can leave it there for 2-3 weeks or more and shake it once in a while, and then you can filter out using a coffee filter. Yeah.
So does the dissolving it in alcohol reduce the antimicrobial properties of it, or do you know how it affects it?
Dissolve. So the more ethanol you use, you mean, okay, so you mean the 75% or
Well, what
50%
I don't
ethanol?
know, just the act of dissolving it seems to me that it would kind of change that molecular structure at some level, right? So I'm wondering if it's going to have an effect on the antimicrobial features of propolis.
It will affect how many antimicrobial compounds you will be extracting out of that propolis. So with water, so like example, if you dissolve it completely and you're using water, let's be extreme here, and you use water, the number of antimicrobial compounds that you will extract are much less. Most antimicrobial compounds will attach to the ethanol much better than they will attach to water compounds. So they will be able to go from that solid phase, that resin phase, into the liquid ethanol phase.
Yep.
So if you use ethanol, it just means you will be extracting more compounds than if you dissolve it completely to a, let's say, a water phase.
So it doesn't
Does it
denature
make
the
sense?
it doesn't denature the compound itself then. It just, it actually keeps the compound, but it dissolves the binder or something. I'm not.
Yeah, it does not, it will not denature the compound. No, it will just mean some compounds will not be in that solution if you do
I
not
see.
add ethanol, because those compounds are not water friendly. Like they do not go, they will not attach to water. So they will not be dissolved in the presence of water. They will not go from one phase to the other. Like they will not be extracted with just the presence of water. But if you use ethanol, then basically that compound will be able to dissolve from the resin and go into the ethanol phase, ethanol solution.
What about extra virgin olive oil at low temperatures?
Extra virgin olive oil, I'm not sure if olive oil would be able to extract many of those compounds. I do not know the answer. My guess is that you would still be better at doing an ethanol extraction first and maybe dissolving with olive oil after, just to be able to extract the most compounds from the resin, than using straight olive oil. But don't quote me, like, do some reading about it. I'm not entirely sure.
Do you know any negative effects of heating the propolis to combine it into a larger mass? Will it deactivate or reduce the efficacy of the antimicrobial nature by heating it?
Because mostly, so when we're talking about heating and denaturing, that is mostly a problem with proteins. So antimicrobial peptides are proteins, and when you heat it up, that molecule will denature and loses its original form and function. So it will denature, and you will not have the same benefits of that protein because you heat it up to past its point of stability, and you denature it. Antimicrobial compounds in resin are different. Those are not. Those are usually more temperature resistant. So you could heat it up. I do not know exactly. I mean, if we're talking about 200, 300 degrees, I do not know how stable they are, but it's definitely it's not like a protein that we cannot put past boiling point. Let's say that you're denaturing most of the protein. Those chemical compounds in resin are different than protein structure, basically.
Have you done any tests on comparing the propolis dissolved in alcohol versus propolis deposited by the bees as far as the antimicrobial effectiveness?
No, I have not. I think, let me see if someone, mainly because it is difficult to study it in the lab, the solid resin ball. But I do think there was a study that someone used as in a petri dish and looked at contact from mites and how that contact killed the mites or was detrimental to the mites. I personally did not, mainly for the things that I wanted to look into in the lab. I had to dissolve it to run through the machines that I wanted to study, I guess, the composition of it and things like that. I had to run through a machine, and I had to dissolve it, use it as in a solvent phase. But I think other people have looked at actual pieces of propolis in the lab and the activity of actual pieces of it. Yeah.
For those who are interested, if you're looking for a good source of 75% alcohol, the 151 proof rum is a suitable solvent for that. I was wondering about the microbial challenge and the fungus challenge, and why not a viral challenge? Are they too difficult to do, too tough to handle?
So there was the problem with viruses, and I find that very interesting, is that viruses are extremely difficult to work with in the lab. Very difficult. They are able to survive and thrive in society really well. But when you put that in a lab environment, viruses are hard to work with. And mostly because most viruses, including honeybee viruses, they are RNA viruses. So RNA viruses are very unstable, and it's very difficult to work with those kind of viruses in the lab and be able to keep them alive, basically. I have not, but I know a couple of people that have tried. I don't think they have published papers yet, but one of them was looking at deformed wing viruses. And so it's an in vitro study. So they have it that in a lab culture, and they're looking at how much the virus, how long the virus survive in the presence of propolis. And again, he didn't publish that yet, but when talking to him a while ago, I remember him mentioning that when the virus was present, he couldn't even get the virus to grow. That was very difficult. But when there is no propolis, no resin, he could get virus to grow in vitro. But because it's so difficult to work with viruses, I'm not sure if he was able to publish yet or able to repeat that, just because of how difficult it is to have replications with that.
And there are so many viruses.
Yeah. So someone asked why some bees are more effective at foraging for propolis than others, and do we know why they would? Resin collection is a genetic trait, and as many other genetic traits, it's inherited. It's not a very high inheritance, if I'm not wrong, but it is a genetic trait. So it means that some bees may just have that in their genetics. And they, for example, Africanized bees, they are much better at collecting resin than our European bees. So if you have some, let's say, Africanized bees, you may see that those collect more resin than your Italian colonies, kind of thing. Is this a sign of better genetic? My experience is that more aggressive hives gather more propolis than docile hives that are more focused on gathering honey and maintaining brood size. Primarily, is it related to a feral trait? I wouldn't say necessarily on its own is a sign of better genetic. You have to look at the whole picture. But because I believe that it's a really good part of their defense mechanism, I think most often it will be a sign of a better genetic. And if they're good at defending themselves against pests and pathogens and parasites, that colony will likely be stronger and healthier. Maybe your experience with those colonies being more aggressive could be related to them having more of the Africanized genes. So that could be that, and compared to the other colonies. Is that related to a feral trait? It could be, because feral colonies do collect more resin, but I wouldn't have any studies to back that up, to make that my assumption a fair statement. The unanswerable question is, to what degree is the propolis source important? It seems one source might not necessarily have the same biological properties as other sources. Brazilian green propolis is from different plant source that what she studied. That is correct. So I studied propolis collection and deposition in Minnesota, and those are mostly popular from poplar trees, not the green propolis. So that's usually a yellow, depending on the poplar tree. It could be the yellow, so cottonwood, or it could be a red propolis. And yes, it does depend on what that propolis source. I wouldn't say the propolis source, but the resin source. Many different trees will produce, even within the same species, you will find different resin chemical composition, because that, again, it depends on how that tree produced that resin. Like I mentioned at the beginning, the tree will produce resin prophylactically. to protect the young leaf buds. But we will also protect that part of the tree that has been attacked. So that's an induced resin synthesis. And that resin, because was induced because of a specific pathogen attack, it will be different. It might be completely different than the same resin produced by the same tree in the young leaf buds. So the resin can differ dramatically, even within the same family, and it will be different in what antimicrobial compounds it carries. So it will be one better than other for a particular pathogen kind of thing. It's very complex.
When I think of a tree that produces resin, mainly I think of things like pine and cedar and redwood. Do those come into play at all with the honeybees?
You know what? One interesting thing, and that can be different in another part of the world, but is that pine trees produce an enormous amount of resin. And I had bees right next to pine trees, and they did not care about it. There was very, very few bees in those trees. They were mostly collecting from poplar, from cottonwood poplar. And that was very interesting, and I don't know why, but pine trees have a lot of resin, like they're dripping with resin, and the bees didn't collect much. But I don't know.
I'm amazed at the comfort level that you have with AFB, because as a beekeeper, or knowing beekeepers around here, they're pretty freaked out about that stuff, and it's surprising that you can keep your lab at least clean in areas and not clean in areas when you're actually spraying that stuff under pressure. Seems like that would just get everywhere.